A Carnegie Institution for Science audio press release for November 16, 2015. Plant metabolic protein tailored for nighttime growth. Everyone who took high school biology learned that photosynthesis is the process by which plants, algae, and select bacteria transform the sun's energy into chemical energy during the daytime. But these photosynthetic organisms activate other biochemical pathways at night, when they generate energy by breaking down the sugars, starches, and oils that they created during the day. New work that focused on this nighttime growth found a protein that's necessary for it to occur, and surprisingly, the role of this protein was linked to the construction of the plant's cellular membranes. It is published by Proceedings of the National Academy of Sciences. The paper is from an integrated international group of plant science experts led by Carnegie's Arthur Grossman, and including lead author Wenqiang Yang and co-author Martin Jonicus, both from Carnegie. It demonstrates that in the photosynthetic algae Chlamydomonas, the protein ferredoxin 5 is critical for growth in dark and for proper membrane organization. Ferredoxin 5 is a member of a family of small iron-sulfur proteins that provide the electrons needed to drive various metabolic reactions in the cell. Photosynthetic organisms typically have multiple different types of ferredoxins, although little is known about how their functions differ. Chlamydomonas has several ferredoxins, including the ferredoxin 5 protein described in this study. The work presented suggests that the ferredoxin variants, including ferredoxin 5, may each have specialized metabolic roles in the cell. The team found that a specific mutant lacking the ferredoxin 5 protein was unable to grow in the dark, in the nighttime, or perform normal metabolic functions, but it had no trouble growing during the light, in the daytime. As it turns out, the mutant algae also exhibited aberrant organization of cellular membranes when maintained in the dark, but did not exhibit them when maintained in the light. These membranes are made up of lipids, which are fatty acids linked to a glycerol backbone. The ferredoxin 5 mutant shows marked alterations in the bond structure of many of these fatty acids, which changes the degree of saturation in the fatty acids present in the membranes. Fully saturated fatty acids have single bonds between all of their carbon atoms, while unsaturated fatty acids may have anywhere from one to several double bonds between their carbon atoms. This in turn appears to impact the lipid composition of the membranes and results in aberrant membrane structure that impairs metabolic processes, including photosynthesis, that occur on those membranes. The team's findings suggest that one of the biochemical reactions the ferredoxin 5 drives through its electron-donating abilities is this alteration in the saturation state of fatty acids. This result indicates that when the algae is in the dark, ferredoxin 5 specifically donates electrons to enzymes that specialize in desaturating fatty acids. It may also donate electrons to other processes in the dark. In the light, however, another member of the ferredoxin family may take on this responsibility, which is why the aberrant membrane structure is only observed in plants that are maintained in darkness. Grossman said this work homed in on the role of ferredoxin 5 and factors critical for photosynthetic organisms to switch between daytime and nighttime metabolism. This image shows ordinary Chlamydomonas on the left and ferredoxin 5 lacking mutant Chlamydomonas on the right. Grown in the light, the mutant is fine, but grown in the dark, the cellular membrane is disorganized. With the independence for nimble pursuit, Carnegie scientists tackle some of the most profound challenges in modern science, fundamentally changing what's possible for us, our planet, and our universe.